So good afternoon, everybody. My name's Rachel from Bats Without Borders, for those of you who are just joining us. I just want to say thank you very much for being part of our very first webinar. We're delighted to have Professor Nancy Simmons as our very first speaker. Just to let you know that this session is going to be recorded and you will be able to get a copy of that later. Before we introduce our first speaker, I just wanted to let you know that next week we're going to be hearing from Dr. Hernani Oliveira, and he's going to be talking to us about the Brazilian savanna, which is a biodiversity hotspot with over 120 bat species. And we think there's quite a lot of lessons that we can learn from Brazil and to Africa. And as I'm sure you're all very, very excited to hear from Professor Nancy Simmons, who's internationally renowned evolutionary biologist, and she is based at the American Museum of Natural History, and she's going to be talking to us today about what we know currently about bat diversity and an update of what's been happening lately. So handing over to Nancy now. Thank you.
Okay, well, thanks to all of you for, I'm trying to share my screen. There we go. For joining today. What I want to talk about today is bat diversity in general and African bat diversity specifically. So I'm going to start sort of with a more global perspective and then focus in on Africa as I move through the presentation today. So, as many of you know, the question that I get asked probably more often than anything is how many bat species are there in the world? But for researchers, I also get very particular questions about what species and subspecies are valid. What are the known geographic ranges? And what is their conservation status? So the reason for this dates to this volume, ultimately, Mammal Species of the World, which was published in 1982. And the author of this chapter was Carl Koopman here on the right, who was my predecessor at the American Museum of Natural History and a bat taxonomist. And in 1982, Carl recognized 918 bat species. Well, the second edition of this book came 10 years later, and this was Mammal Species of the World, Volume 2, 925 bat species at that time. So I think 7 or 8 bat species have been added in the meantime. Well, Carl passed away, unfortunately, in 1998, and I took over the bat chapter. And at this point, I thought it really wouldn't be that much work because I had not just inherited, in essence, Carl's position at the American Museum, but I'd also inherited his personal copy of the previous edition of Mammal Species of the World. And he'd written little notes in the margins, scratched things out, written little added in species. And I thought, well, you know, here Carl's already done most of the work for me, so it won't be that hard. Well, I was wrong because by 2005, there were 20% more mammal or bat species than the previous edition. So interestingly here though, if we look across these different snapshots in time of what we understand about species diversity, the percentage of mammal species in the world that are bats has stayed pretty constant through time. So even though we've been recognizing increasing numbers of bats, in context, we've also been recognizing increasing numbers of other mammal species. So roughly 20% of living mammals are bats, living mammal species are bats. So in this volume, familiar to many of you, there's an account for each species. So it has the species name, authorship, and so on, all these data which are of interest to specialists. However, this is all now 15 years out of date. So to put that in context, this is a picture of my younger son the year that 2005 volume came out. And this is what he looks like now. Basically, a lot of time has passed, a lot of changes. So in the world of bat taxonomy and diversity, there's been a tremendous amount of new information produced. And starting in I guess it was just before the publication of the last volume, I realized I didn't want to ever have to sort through all of this in a huge pile again. And so with the help of Andrea Cirinello, we decided that we needed to move this into some sort of database format. And so at that point, we started keeping a flat file, Excel file, adding new species, moving things around, and so on. So how many bat species are there in the world now? Well, as of April, our last count, last month, 1,421 bat species. So this is over 300 more species compared to the last edition of Mammal Species of the World, a 27% increase. So where have all these things come from? Well, worldwide, about 22 new bat species have been added to the list per year over the last decade or so. Of these, about 60% are new species. So these are taxa which need new names because there's no pre-existing name for them. However, 40% are raised from synonymy. That means they're subspecies. Most commonly, it's that researchers have discovered that something that was previously recognized to be a subspecies is now, with more information, we realize that they're actually distinct taxa, although sometimes they're surprises, as we'll see later. Well, where are all these new findings coming from? Well, sometimes it's from new fieldwork, new discoveries. Someone goes out, puts out a bat net in some place that hasn't been surveyed before and catches an animal that nobody's seen before. However, often these days it's new technologies which are giving us more information about species diversity, particularly using acoustic survey methods. So bat detectors really changed the world for bat biologists because suddenly we can listen to the night sky and in some cases determine that there's more species out there than we thought there were originally. And by going back and using DNA sequencing data, we can find that we can evaluate whether the things that we may have captured, we think, is this the same, is this different? We can use sequencing technologies to try and resolve those problems. So the combination of new field work, using acoustic data, and going back looking at old museum specimens in some cases, and new samples as well with DNA sequence technology has really changed what we know. In addition, study effort plays a large role. I decided a few years ago to try and figure out just how many papers on bats there were out there. So what I did was I went and looked for the number of papers published per year, averaged over a course of several decades, and using Google Scholar. And this is what I found. So back in the early part of the last century, 20 to 25 papers per year were published on bats. And this continued to go up to the time I started my career working on bats in the early 90s, and there were about 550 papers published per year throughout the 90s. But then something remarkable started happening. It just started going up and up and up. So since 2010, there have been over 2,000 papers published per year on bats. So, and this is just continuing to increase. So if we look at 2019, which is the last year that we can assess a complete year's amount of research on bats, we find that there are around 3,500 papers published on bats last year alone. If we break this down by sort of different topics, these are the search words I use. Chiroptera and ecology, 2,400 papers. Chiroptera and conservation, 2,000 papers. Chiroptera in Africa, over 1,000 papers that included bats and Africa in that paper. So this is a huge amount of literature. But what does it mean for our understanding? What are the taxonomic diversity changes that we've come to recognize as a result of all of this work? Well, let's focus in on Africa first. The number of new species added to the list since 2005 is really substantial for Africa, and I've pulled Madagascar out here separately. 67 additional species in the last 15 years, 20 for Madagascar. How does this stack up to other regions of the world? Well, I put the other numbers on here. The general trend that we see is that in tropical regions and regions which had lots of unexplored territory, the numbers are much higher. So North America, only 2 new species since 2005 recognized. South America, 80. If you add Africa and Madagascar together, that'd be 87. Southeast Asia is another big hotspot with 69 additional species. In terms of overall totals, these are the numbers. What we understand today is over 300 species in Africa, so 326 as of our most recent count. This is second only to Southeast Asia with 388 species at present. And of course, how you define these geographic regions depends on exactly how you get the count, so these are just sort of rough estimates. So let's focus in now on what's new in the African region. Well, one of the big differences or new big changes that I find very exciting is that there's a new family of bats in Africa since the last edition, Rhinonycteridae. So this is an old name that dates back to Gray 1866, but was only used as a tribal level subdivision before now. However, now it's been elevated to full family level. So this happened very recently in 2014. Foley et al. We're doing a phylogenetic study of rhinolophid and hippocidera bats and came out with some surprises. So here is their phylogenetic tree, and most notably here is they found that the group now called the family Rhinonycteridae here is as different from other members of Hippocideridae as both of these are from the rhinolophids. And so this is why they decided that this group deserved family level recognition. So who are the Rhinonycteridae? These are the trident bats. There's 10 species now in 4 genera. They're found in Africa, Madagascar, the Middle East, but the majority of the diversity in this group is in these 3 genera, which are from the Africa-Madagascar region. There's only 1 species, Rhinonycteris, which is from outside this area. Here are some lovely pictures of these animals. This is the African trident bat, and this is where they take their name from, or this incredible nose loose with these 3 prongs at the top. And these are the same species, by the way. There's some incredible polymorphism in color variation in these animals. They're just lovely critters. And I have to put in a confession here, which is I have done no personal field work on bats in Africa, which is something that I'd really like to remedy at some point. I just love to get to see these animals alive personally. But in the meantime, I just have to be satisfied with pictures. Okay, moving on. Hippocideridae, the Old World leaf-nosed bats, is another very diverse group in Africa. There's currently 90 species in this family, spread across 7 genera. And there's recently been 2 new genera added for Africa. And this is again as a result of recognition of lineages being more distinct than we thought they were. These are the 2 new taxa, or 2 taxa which are now being recognized as distinct genera. These are old names, obviously, but they were in use. They were considered junior synonyms until very recently, Doryrhina and Macronycteris. So who are these guys, and where did they come from? Well, another paper by the Foley et al. Group in 2017, and I love the title of this, so I need to put it up, Towards Navigating the Minotaur's Labyrinth, Cryptic Diversity in this group. Hippociderids, and there's a number of groups that have a great deal of diversity. We're only now beginning to sort out. So Doryrhina, this may be familiar to many of you working in Africa. We used to call this animal Hippociderius cyclops. Now it's Doryrhina cyclops. Macronycteris, another big change. For those of us who've been saying Hippociderius commissionii or Hippociderius gigas for years, we have to learn to use a new genus name. Macronycteris is now the correct name for this clade of just gorgeous animals. So this is the reason this group is now recognized as distinct. This is a phylogeny from Foley et al. And here we have the clade including Macronycteris and Doryrhina. As you can see, it is very distinct from the rest of Hippocideridae, and most notably here was their finding that these other genera are actually more closely related to Hippociderius sensu stricto, which is why these guys need a new name. And also just in terms of overall diversity, in the African context, these circles here indicate species which are African. So this entire clade, the Macronycteris plus Doryrhina clade, is African, whereas Hippociderius is largely an Asian group with African taxa within it. However, it's not just molecular data that are the arguments for recognizing these guys as different and in important ways. These are nose leafs of Macronectarus and Dornirhina. The nose leafs here on A, B, and C, these are various species of Macronectarus, and you can see how similar they are. Over here on the right, D, is Dornirhina cyclops. So the distinctive, the differences that we're detecting with sequence data are often also reflected in morphology, and that's really an important point, I think. Most recently, just published a couple of weeks ago, is another huge revision of Afrotropical Hippocidarids by Patterson et al. And this study included massively more data than previously analyzed. So I'm going to show you some of their results just in pieces here. So this is the bottom part of the tree. They found strong support for Macronectarus being recognized as a distinct genus, and also for Dornirhina. But interestingly here, one of the species previously left in Hippocidarius turns out to group in Dornirhina. So actually there's two species in this genus now. There's Cyclops and Cameroonensis. Now moving up the tree, we're passing now through a section of Asian taxa, but I want to just sort of keep going into the rich African realm here of Hippocidarids. Now I understand most of you probably can't even read the names here, and that's fine. I just wanted to make the point that when we looked, when Patterson et al. Group analyzed these sequence data, this was a mitochondrial gene tree. They found some remarkable things. For instance, multiple lineages, Hippocidarius ruber 1, 2, 3, 4. In Kaffir, they recognized 8 different mitochondrial lineages. So this is mitochondrial data, so it doesn't necessarily imply that these are distinct species, but some may be. And it really suggests that there's a lot of unrecognized cryptic diversity here. And I don't want to go down the rabbit hole of presenting lots and lots more trees. They also analyzed nuclear data, and the conclusion by looking at both these mitochondrial trees and their nuclear data is that there's now compelling genetic evidence from both mitochondrial and nuclear sequences for several additional unnamed species in Hippocidarius. And I want to point out here that this is sort of the going trend when people look at complex groups across whole continents. The discovery of cryptic diversity is just an ongoing process. Whenever anybody looks closely, they find new things. In this case, the Patterson et al. Group argued that conflicting appraisals must be adjudicated by large-scale integrative analysis of echolocation calls, quantitative morphology, and geometric morphometrics. And I love this approach because bringing in the morphology, the echolocation calls, and so on into these projects greatly enhances our understanding of the diversity and really what's there in these lineages. So this approach needs to be applied across the board, not just in Hippocidarius. And I believe there's a lot of additional species that are going to be showing up over time in other lineages as well. Then you get into the whole name problem. And for taxonomists, this is a big deal. This is a map from the Patterson paper. And this shows the localities for all the the holotypes of named forms of Hippocidarius in Africa. So when you come up with a study showing that there's cryptic diversity out there, then you have to sort out how these various genetic lineages match up with the taxonomy. And that is often a big deal. There's a lot of names here, and doing this last part, sorting, putting the right names on the right lineages is a huge challenge. Okay, back to the bigger picture again. I said there was a new family of bats for Africa in the last 15 years, but actually I lied. There's really 3 new families of bats in Africa. There's 2 more here I want to talk about, the Cistugidae and the Miniopteridae. Let's start with Cistugidae. These are the wing gland bats. And this family was elevated to, or this group was elevated to family level by Lack et al. In 2010. There's 2 species, and they're endemic to Africa, to southern Africa. The take home from the Lack et al. Paper is really one big thing, which is these are not Myotis. These 2 species for decades have been either recognized as species of Myotis, sometimes in their own subgenus, but in this paper in 2010, these researchers came up with very convincing evidence that Cistugo does not belong in the genus Myotis. So here it is in their evolutionary trees. Cestugo out here. And what's notable in this diagram is here's the group, the Myotis group, which Cestugo was placed in. But not only does it not go in Myotis, it's actually outside the group that includes all other Vespertilionids, and here's some other subfamilies of Vespertilionidae. Cestugo, long branch links here. These are very different in evolutionary terms from the group in which they were previously classified. So they actually look a lot like Myotis, but in an evolutionary sense, it's a completely different lineage. And this is a group which is endemic to Africa. Now moving on to Miniopterus here. This is another group which has been raised to family status recently for the same reasons. It does not belong with all the other Vespertilionids. It is clearly very distinct, falling outside this group. It's a sister group to Cestugo plus the other Vespertilionids. Who are these guys? These are the long-fingered bats or bent-winged bats. There's a really gorgeous one here. There's currently 34 species of these guys. And they were raised to family level by Miller Butterworth et al. In 2007. This name is more familiar though because it had been used as a subfamily name for a long time prior to its becoming its own family. More interestingly though, from the African perspective, is just the view of diversity of these animals. So in 2005, there were 9 species recognized in Africa and Madagascar together. Now there are 19 different species. So who are some of these? Well, the Mozambique long-fingered bat was described in 2013, Miniopterus mozambicus. Here's another one this time from Northern Africa, the Maghrebian bent-winged bat, described in 2014. Here's Allen's long-fingered bat, described in 2009 from Madagascar. And I could really just keep going. Almost ad infinitum here, 2015, 2009, 2011, 2010, 2009, and so on. So to avoid making this a nonstop talk of just new species after new species after new species, I sort of want to go on now and talk about the bigger patterns again. So lots of new species since 2005. How do we track these things? So I gave you this view earlier of how much is being done in terms of work on bats. Well, 2019 alone, there were 1,500 papers on bats and taxonomy. So just tracking this stuff is really been a nightmare in some respects. So going back to this tracking problem, I work with Andrea. What started out as piles of paper in my office eventually became a flat file data set in an Excel file that we emailed back and forth to each other. But we're trying to move on from that. However, we've needed help in doing this. So starting a few years ago, we realized that we really wanted to have a community effort to stabilize bat taxonomy. And so we formed the Global Bat Taxonomy Working Group. So this is a working group now, which is under the umbrella of the IUCN Bat Specialist Group. So we have a formal affiliation with the IUCN. Founded in 2017. With the goal of a community-based approach to updating and stabilizing bat taxonomy. Currently, there's 18 members of this working group, and it's still growing. We're adding people all the time. This is where people are based. We're sort of heavy in North America and Europe because that's where a lot of bat taxonomists are, but we're trying to get people from throughout the world who are working actively in bat taxonomy to help us out. So currently, we have 2 African members, Paul Wabala from Masai Mara University in Kenya, and Aram Monajem from University of Eswatini, who are members of this working group. And so we work together when there's controversial problems with recognizing diversity, whether it has to do with details of taxonomy, details of publication, just trying to make sure that we get all the right names on all the species. So where do we go from this old school bat volume on your desk? Well, we're trying to move into the modern age with cloud computing and databasing. So our big step forward was establishing this website a number of years ago, and it just went live, I guess in 2018, it might have been 2019, batnames.org, which looks like this when you log in, and any of you can do this. And I am now going to try to let's see, get out of this so that I can, let's see. Nope, that's not what I wanted to do. Nope. Ah, there we go. What I want to do is give you a live presentation of what this website can do. So now I am in the browser online actively. So when you log into the Bats of the World website, you have a number of options of things to do. So one way to get to data is to use the Explore function up here. And in Explore, you get to a higher level taxonomy of bats here. Let's go to one we talked about already today, the Cystugidea. So if I click on that, here is an account for that family. It tells you how many species are in it. It allows you to scroll and see there's comments for each family. There's distribution information.
Nancy, sorry to interrupt you. We can just see your PowerPoint presentation. If you just share your website. Sorry, I know that's a fault. It's a problem with Zoom. It's a problem with Zoom. Thank you.
Okay, let's see. All right. It says I'm screen sharing. Okay, what are you seeing now?
We're just seeing your in PowerPoint, but the escape mode. So I think we just need to, I think when you maybe stop sharing and share again, and then just share your browser rather than, there we go, yeah.
That. Okay, now are you seeing the Bats of the World website?
Now we're seeing you.
You're seeing me. All right, now I have to share again, I guess?
Yeah, so share again, and it should give you an option of which view you want us to see.
Thank you.
There
now
we
what
go.
do you think? Okay, perfect. Okay. All right, so that's the world's website. Now we're in the real thing, and hopefully you'll see this now. Yes?
Yes, we can. Thank you.
Perfect. Anyway, so what I did was from the front page, you can go to Explore, and then you can see the higher level classification. You go down and click on anything you want. Here I've clicked on Cystugidea, and it shows the authorship, the number of species, included genera, distribution, and so on. And a really cool thing here that I really value, but it's taken a lot of time to implement, are the references. We actually have direct links built in to the references so that you can click on those and go straight to the paper. So here I am slowly loading up the Lack et al. Paper. So anyway, this is hopefully a tool that many people will find valuable for their research. But let me show you a little bit more going through. Let's see, we want to find out more about a genus. We can just click on Cystugo here, and it takes us to an account for that genus. And then we can scroll down, and there's accounts for each family. These include all the basic information that was originally there in the Mammal Species of the World volumes, plus it has the references with the links. So now I'm going to go back to the front page. Another feature which is highly useful, I believe, at least I use it a lot, is the query bar. We can type in anything we want, and it will help you with spelling and so on here. I'm going to go to Megadromatidae because that's a group I haven't had a chance to talk about yet, which is found in Africa. Here we go. We have our in gray, we have something on the family, and we have various accounts here for species. Now, one thing I haven't shown you yet is that there's also maps involved here. For each species account has a map, and we can zoom in and find out where those actual places are. This is downloaded from GBIF, so you have actual locality data here for all of these things. And if you want to, you can go into GBIF directly and find information on every single one of these points, where that data came from, what collection, or whatever. So these accounts are what we call long accounts, which are the accounts where we've actually done all the work that we hope to do in updating. So it has synonyms present, it has all the references and so on. Unfortunately, we haven't had time yet to update everything to that level. So to show you an example of a group that we're still working on, this is the genus Rhinolophus. For some of the more speciose groups, all we have are, in essence, lists of what the valid species are at this point. We don't yet have, at least live online, all of that other information. However, it's all behind the scenes. So if anybody ever needs information, if you click on something like this, and it's like, oh, I really want to know the synonyms, you can always email us and we can provide that information. We're rapidly moving through all these accounts, trying to bring them into that long form so that all that information will be available. There's a submit area in the website here. So if you publish something that you think is, includes taxonomic changes or range extensions or anything you want to bring to us or to our attention, you can email it through us here. And down here at the bottom is the information on how to cite the database. So now I'm going to stop sharing again so I can go back to my PowerPoint presentation. Since now we know we have to do it that way. There we go. Okay, back into the PowerPoint. Moving on. The last thing I want to talk about briefly doesn't have anything directly to do with African bats, but I want to bring this to everyone's attention as you're all obviously interested in bats since you're here in the first place. And that is a new organization called GBatnet, the Global Union of Bat Diversity Networks. So this was effectively born last month. However, it has a long history and in fact actually originated with discussions among the leaders of a variety of bat research networks and conservation networks that took place in South Africa in 2016 at the International Bat Meetings. And at that time, leaders of various networks around the world realized that there's not enough interconnection between bat groups in Africa, say, with bat groups in North America, with genomics groups, with disease surveillance groups, all these different pre-existing networks of researchers. There was no centralized way for people to talk to each other. So we started talking about developing a global network of networks back then. But it wasn't until the coronavirus outbreak, frankly, that we were driven to sort of mobilize this quickly. With the pushback against bats in general, with a lot of, frankly, wrong information out there in the world about bats and links between bats and viruses, with little attention to the importance of bats and ecosystems around the world, we decided that it was really time to get GBatnet up and running to help counter, basically, the bad information that was getting out there. So GBatnet now has quite a presence in social media. Here's the front page from our Facebook page and also from Twitter. And this is to remind me to explain again that GBatnet is not a new organization. It is well, in terms of being a completely different set of people, what it does is simply brings together existing networks that are working on bat research, conservation, and sustainability. So some of the things that we do is, through our social media outlets, is post good links to good articles about bats, which are scientifically accurate and tell stories that we want to make sure get out there. We can also share through this network information on funding opportunities that maybe people aren't aware of. We can advertise back and forth through the community various conferences and summits. This one was just posted a couple days ago. This is the World Bat Twitter Conference, and it's not too late for any of you who want to join this. The abstract deadline is May 17th, so that's coming up here in just a few days. But anyway, this kind of information is we can help spread this through this network. We can also help with people who are looking for help. Here's one I just found this morning from the African Bat Conservation Network, looking for help for a variety of things. And finally, we can circulate important documents, important information. For instance, this just came out. This is the recommendations of the Eurobats Advisory Committee, basically on research and handling bats. So these sorts of things, this GBatnet network gives us a way of really rapidly spreading information around the world amongst bat research networks. In addition, we're working to generate some new content to get out there, scientifically accurate content. So there's a blog that we've established. And we've been soliciting articles by experts, basically summarizing scientific literature and making it accessible to non-experts. For instance, this is one of the first articles here, Human Impacts and the Spillover of Zoonotic Diseases. Here's another one. Killing bats won't help prevent disease, but here's what will. And these links here, which are shown, I think the colors have been updated subsequently to be a little nicer. But anyway, these the things shown in yellow here are actually links to scientific papers. So if you want to read about bats as special reservoirs for zoonotic pathogens, or want to read about transmission of zoonotic diseases here, you can click on those links and get straight into the scientific literature. Many people are interested in knowing how they can participate in GBatnet. Well, there's a number of different ways. The easiest is to simply link up with us in social media. So through Facebook, which is sort of my preferred area because I'm old. Twitter, though, I'm learning how to use Twitter. We have presence on both. You can follow us on Twitter, join us on Facebook directly. However, I do want to stress that GBatnet is really a network of networks. And so the best way to get involved in research, conservation, education, and so on about bats and help participate at this large-scale level is to work with your local organization. So in Africa, that would be Bats Without Borders or the African Bat Conservation Group. Depending on where you live, there are, and what your research interests are, or educational interests, or whatever, there are different networks that you can work with. And we're trying to bring all of those into the GBatnet umbrella, in essence. Not to tell anybody what to do, but simply to help build links between all of these really functional, wonderful groups that are doing a huge amount of valuable work. So that's where I'm going to wrap up my presentation. I want to one more time say thank you to Andrea Cirinello, without whom the Bat Names database and all of this tracking of species diversity would never have been possible. If this was a scientific talk, she would be a co-author for sure because of her vast amount of work on this. Also, photo credits. I have liberally snatched photos off the internet, off iNaturalist in various places that were taken by these people. Some of them know that I'm using their photos, some don't. Anyway, I just want to make it clear that these pictures all were taken by other people who went to a lot of trouble to get them, and they're really appreciated. So anyway, that's the end of my formal presentation. So I'm going to stop sharing my screen there, and we can have some questions.
So thank you very much, Nancy. And just to remind you, if you want to ask any questions, to please post those in the chat. And just to also let you know, Paul Wabala has been trying to join desperately. He's been messaging, so he sends his apologies. So we have a couple of questions already. So could you give a brief about theropodid diversity in Africa?
Oops. So, theropodid diversity in Africa, it is very much the same thing as the stories that I told about hippocidarids and so on. As people look more closely, they're finding more taxa. I think one of the groups that is most problematic at present is Epimaphorus. It's still unclear how many species there are there. There's more work being done on that group. Again, it's a moving target, and one of the things I've had to become used to and just get over, I guess, as I've been working on bat taxonomy all these years, is the idea that we would sort of ever be finished, because every time we look more closely, we learn more things, and as new tools are developed, we're learning more about the complexity of evolution in some of these groups, the gene flow between species. When is something, when are evolutionary lineages distinct? How different do populations need to be? How do they share genes? It's a moving target. So yeah, I would say in theropodids, it's looking at Epimaphorus especially. There's more taxa there, I think, that are coming.
Thank you. And then we've got another question. How do you reconcile nuclear or mitochondrial DNA data with other types of data that you mentioned, such as morphology or echolocation calls, if they are at odds in terms of the place of the species? And does each method have a weight, or is it more case-by-case basis?
There's no single answer to that question. I would say that in general, when people are doing integrative analyses, so you have maybe mitochondrial data on one hand, you have some nuclear data, you may have echolocation call data, you may have morphological data. What we're looking for is sort of the best guess scenario for what is most likely to be the correct answer for the phylogeny. And so when multiple different lines of evidence point towards the same answer, that's the kind of answer we want to accept. So I'm much more likely to accept a hypothesis that two things are different species if there's multiple lines of evidence supporting that. When there's incongruence between, say, mitochondrial nuclear data, what's most often the case is that due to female phylopatry, where females stay home and stay in smaller ranges and males are the ones that disperse, we often find that there's more structure to the trees based on mitochondrial data, and we know that because if there's that structure is not present in the nuclear genes, that usually means that the males are doing the gene flow. So even though there's a lot of structure in mitochondria, it may still all be one species with the males doing all the moving around. But in a case like that, sometimes it's just the nuclear data the nuclear genes just don't evolve fast enough, so it may be that we don't really have the right genes. But if there's no other support for two things being different than mitochondrial data and the nuclear data show no structure, then it's probably not that these are different species. But if there's a distinct morphological difference as well as mitochondrial difference, then we might want to accept that as two different species. So these are the complex problems where you have to look case by case and look at the quality of the data, look at the sampling, and are there big geographic gaps in the sampling before reaching a final conclusion. So there's not really one size fits all. And different researchers have different comfort levels with recognizing something as distinct or not. And that's part of the reason that we need this global bat taxonomy working group, is we need to have more than one person making the final decision on some of these controversial cases.
Great, thank you. And we have another question. Taking into account the undescribed species of bats that are still being discovered, how many more species do you think we're going to be added to the current world list of bat species? And also, we have a lack of taxonomists in many groups of animals worldwide. Is that true also for bats? And if so, how many more taxonomists do you think we need to do a good job of describing undescribed bat species? So there's two quite tough questions for you.
Okay, I think I can break that down. How many bat species are there really in the world? I have no idea. I am convinced we're going to go over 1,500 in not very many more years. Will we get to 2,000? I guess I would be surprised if there were that many unrecognized bat species, but I think there's still a lot out there. I mean, the pace of finding new ones, so to speak, has not slowed down at all in recent years. Are there enough bat taxonomists? Well, actually, we are lucky that there are a lot of bat taxonomists, and particularly there's people working in, increasingly, I would say, in the countries that are biodiverse. So we have young researchers with some lab access in many countries around the world now, and they're taking the leading role in a lot of this work and figuring out, what's new and where is it, those sorts of questions. I think it would be awesome if there were lots more bat taxonomists. It would just make our work easier and we could move faster towards figuring out the real diversity out there. But we're not, thankfully. Bats do have a reasonable number of people working on them. Also, I think that back when I first started working on bat phylogeny and systematics, there were very few people who were doing it because a lot of people were actually more interested in ecology and population genetics and things like that. And I think those really are, really fascinating areas of research. But now it turns out, you know, that people can do all of these things. And so by helping enable people who are working on, say, population genetics of groups to be able to learn how to interpret their data in a sort of larger scale context, and by, you know, not trying to tell people that they can't do taxonomy if they weren't trained necessarily to do that. If we help people, then we can see a lot of steps forward that way. So I'm hoping that there'll be more people doing bat taxonomy in the future and looking at diversity.
Great, thank you. So we have a question from Ram Kumar, who's a researcher in India. I was just saying, he was wondering if there's been an updated name for Pteropus giganteus.
That's something you can find out probably for yourself using the bat names database. I would have to go back and look at that. So some of these species that have wide geographic ranges have been split up in recent years, and it just depends on where you are in the range of that animal, often what the proper name is that applies. I didn't use one of my favorite examples today, which is from the New World, where I do most of my own field work, which is Pteronodus parnellii, is a species that was thought to extend basically from Mexico throughout the Caribbean through almost all of South America. So there's specimens all over the place from this range. But recent revisionary studies showed that actually it's a complex of many species, and that the name only properly applies to those bats from the island of Jamaica. So everything else throughout the range of this species, everything in South America, everything in Central America needs a different name now. So sorting this stuff out can be annoying and confusing, but it takes often looking at the history of names across a range to figure out what's going on.
And our next question is, what do you think are the main gaps in African bat research?
Ooh, you know, that's hard for me to say. So we have a lot more records from some countries than from other countries, and it has to do with the history of research in those areas. Some of this is just the colonial history of Africa, where you know, people went and made large collections in certain areas, but not others. Some of the important gaps are in sampling of some taxa that extend, particularly West Africa, the connections between collections from West Africa to East Africa. There's some gaps in the middle, and then connecting that up to the north. I think that more field work in some of these areas, obviously some of them are not particularly accessible, would be really helpful. There's also some gaps as you move down, you know, through Angola and places like that, where we don't have a lot of material. But it varies according to what taxon you're working on. I've been working in Myotis lately, so I have a better feel for sort of where the gaps are in Myotis. And I had a student who was working on Eidolon, and so I have a picture of Eidolon, you know, map in my head and thinking, oh gosh, I wish we had samples from up there. It just really depends on what taxon you're looking at, where the gaps are. I'm not sure if that helps much, but.
Yeah, thank you. And is there much work being going on with looking at historical museum records in terms of now, you know, advances in molecular techniques to kind of revisit, you know, historical records and update them, but also to share that data?
Yeah, so I think in many of the trees that I'm showing you, in many of these studies, some of those records are from museum specimens. So particularly that gigantic mitochondrial tree I showed you of Hippocyterus from Bruce Patterson's lab. When you're looking at mitochondrial genes, it's quite, well, I won't say easy, but relatively easy to get useful sequence data out of museum specimens. And so a lot of mitochondrial studies these days do include museum specimens. So at the American Museum, we're routinely getting requests to sample older samples to put in, because in some cases they come from places that nobody's going to be able to go back to anytime soon. And so being able to place those historical records into context is really important. It gets harder when you're working with nuclear genes, because you can't necessarily, because there's only one nucleus per cell. And if the DNA is degraded because it's an old museum specimen, you may not be able to get any results out of it. So people tend to focus on trying to get mitochondrial genes out of museum material. And this is just being done, increasingly. And I think that the more we can look at and make use of these old museum records, the more we can flesh out a lot of what we know about diversity patterns. And even, I talked today about just what is, what are the lists of species and how many there are. But really, in many ways, the much more interesting question is what is the distribution of this diversity across the landscape? And often the only way to look at that is to make reference to museum specimens.
Thank you. So just to let everybody know, we only have a couple, about 5 more minutes left. So I've got 2 more questions. So which of the techniques that you've mentioned before, so echolocation, field sampling, genetics and morphology, do you think would be the most useful to invest more effort and money to increase the discovery of species, especially in high diversity species with limited resources? I think
I'm gonna punt on that one and say I think all of these are important. So every researcher has the methods they're most familiar with, but all of these data are useful. We do know that morphology, the traditional way of skin and skull measurements, fur color, and so on, only gets you part way to understanding species diversity. So there are many cryptic species out there where if you hold them in your hand, you can't, are these 2 different species or just 2 slightly different individuals? You can't tell. Morphology doesn't get you all the way, but on the flip side, just doing genetic work also leaves out a lot of important information. So I feel that, that's the reason I put that quote into my talk from the Hippocyterus paper. I really feel that integrative studies, including all of these different kinds of data, are important. And when somebody is describing a new species today, I think you should include all of these components. So ideally, there should be a clear morphological description and morphological comparisons to similar things. There should also be genetic data and echolocation data if you have it. All of this is important information, and so a comprehensive description of a new species is far more valuable than just picking one out on a mitochondrial tree and saying, this is 5% different than that, so I think it should be a different species. Bringing all the data together is what is really informative.
And another question is, many of us on this call probably don't describe ourselves as taxonomists, but we may be sampling unique species. What do you suggest to ecologists in the field interested in contributing to that taxonomy?
Well, one of the things is to contribute to bat taxonomy, you basically have to collect samples. So this is controversial for some people. We cannot do taxonomy if all you have is a picture of a bat. We can't do taxonomy with that. So minimally wing punches, ideally whole preserved animals. So killing bats doesn't make anybody happy, but if you think about whole populations out there, taking 1 or 2 individuals from a population should not be having any effect on that species. So collecting samples and depositing those in some sort of museum where they can be archived is important. Now, it's easy today with the internet to make contact with researchers who are working on particular taxa. So if, for example, you catch an animal and you go, wow, this doesn't fit the key that I have. Is this something different? I don't know. You should be able to go online and find the name of somebody who's been publishing papers recently on that taxon and just get in touch with them. I love it when people email me and say, I think I have a new species of X, and then I can help put them together with somebody else who's working in that area. There's no reason why, and in fact, for most of us, a lot of the new taxa we've been involved in describing haven't been things that we have personally captured ourselves, but they've been things that have been brought to us by other researchers who are the people in the field who are actually working with the animals, and they're the ones who make the new discoveries.
And just to sneak in a couple more, if we have time, what are good morphological characteristics to be looking out for when describing a new bat species?
Well, that depends on the taxon, but standard measurements are really important. Body weight, is important, and forearm length. Those are probably the two most important external measurements, although we also measure a variety, you know, body length, tail length, ear length, tragus length. So there's a series of standard field measurements for bats, which are important. Fur patterns, so colors and also banding of the fur. So whether if you blow on the back of the bat between its shoulder blades, do you see that each fur, you know, the length of each hair, is it all one color, or are there 2 color bands or 3 color bands, and what are those color bands are important. When we're working with voucher specimens, we also do a lot of measurements, skull and dental measurements, and sort of these standard morphological traits are things which you can find in sort of pretty much any modern species description of bats should have a set of measurements, and people take slightly different ones, but if you're like holding a bat in your hand, it's the forearm length, it's the fur patterning, it's the for things that have nose leafs, what is the form of the nose leaf, how many projections, is it fused to the lip, is it not fused to the lip, those sorts of things. So the kinds of characters that you see in keys are the kinds of characters that we use to tell animals apart. So it's 10 o'clock now, and I unfortunately have to go to another meeting, and I know that Rachel wants to wrap things up, so I'm going to bow out now, and I want to thank you all for listening to me this morning.
So thank you very much, Nancy, on behalf of Bats Without Borders and all the participants here today. Really appreciate your time, and great talking to you.